Hello and welcome. I'm Aretha Lafayette, Program Manager for Strategic Research Initiatives in the Office of Research and Innovation, and I'm happy to serve as your MC and guide for today's session. Today's speaker is someone who doesn't just navigate challenges, she thrives in them. She is the medical director and co director of the Clinical and Translational Research Center, chief medical officer of the Texas Biomedical Device Center, and medical science research director of the Center of Brain Health at UT Dallas. A board certified emergency medicine and lifestyle medicine physician, she has over 2 decades of experience in clinical research and translational research. She earned her medical degree from UT Southwestern, where she also completed her residency and later served as emergency medicine faculty for over 23 years. Dr. Wigginton holds a Master's of Science in Clinical Sciences with distinction and has extensive training in study design, biostatistics, and regulatory approvals, including FDA, IDE, and IND applications. She has collaborated on numerous interdisciplinary research projects and has secured NIH and other grant funding. As a longtime member of the UT Southwestern Institutional Review Board, she brings deep expertise in clinical trial ethics and execution. Her work bridges the gap between scientific discovery and real world medical applications, ensuring innovations translate into impactful health solutions. I'm delighted to welcome Dr. Jane Wigginton for her talk, Translational Research Unlocked, where she will share key basic strategies and insights on how groundbreaking ideas become life changing medical advancements. Dr. Wigginton, the virtual floor is yours.
Oh, thank you so much, Aretha, for that lovely introduction. And I'd like to thank the Office of Research and Innovation for inviting me to share in this part one of this very cool and fun clinical trial series. And I'd like to thank all the people who are in attendance for sharing part of their morning with me. I know you all are busy, and hopefully, this will be very enjoyable and productive. So, today, my goal is to share some of the things that I've learned along the path of the translational research and the clinical research and the clinical practice that I've done to try to give you some tidbits of things that may help you get over a hump. And I'm also going to put in a plug now and at the end for the CTRC that's new. And I'm going to show a picture at the end of the website where you can visit us and come and get help with hopefully a project that you're working on. So over the years, I have seen a lot of clinical trials done and worked through. But the very first clinical trial that I ever became aware of that was FDA regulated started like this. I had a friend who was an interventional cardiologist. He still is an interventional cardiologist, and he was an electrophysiologist. And those folks do really incredible, dangerous work. They stop people's heart, they stop bad pathways that are going through the heart and everything. And then hopefully they start their heart again. So he was scrubbing in one day with his partners, and one of them looked over and said, "Hey, did you see the news today? Oh my gosh, there was the craziest story in the whole world." And in that story, they talked about this little boy whose dad dropped dead in front of him. He had sudden cardiac arrest. And the little boy didn't know CPR or anything, but he had seen it kind of on TV before. And he ran to the broom closet and he got the toilet plunger out and he ran over to his dad and he got down over him and used the toilet plunger to resuscitate him. And these electrophysiologists were laughing, going, "What a crazy story. That is the craziest thing I've ever heard. Who would ever think of using a toilet plunger?" And the fact that he got him resuscitated was even crazier. But my friend, who was really brilliant and really attuned to medical things, had an idea. And he said, "Gosh, maybe there's something physiologic about this toilet plunger that helped this little boy resuscitate more than just having hands on the chest. Because when we do CPR and you push on somebody's chest, it pushes the blood out. But maybe the suction cup of that toilet plunger actually pulled the chest forward, which increased the negative pressure in the chest and sucked the blood back in, reloading the heart so that the next time he pushed, there was actually more blood in the heart to push to the brain and the other vital organs. So, what he did was he developed this device based on this little boy and the toilet plunger. And this device shows how much pressure you put down and how much you pull up. And they took it to the pig lab. He had a big pig lab. And he took it to the pig lab, and I actually got to go and work on it with him a little bit and showed that you could resuscitate better with this device than you could standard hands-only CPR. So one day they were in the OR and the patient had a cardiac arrest. And so they were using this rescue pump that was an investigational device at the time. And the anesthesiologist put his thumb partially over the endotracheal tube while they were using this rescue pump. And he noticed that the person's blood pressure went up. And he was like, how does that happen? And what that little bit of thumb over the endotracheal tube or the breathing tube that they use in the operating room did was it actually enhanced the negative pressure that was pulling the blood back in the time that they were not pushing. And so what my friend did was he developed this little device that went on the breathing tube and it occluded it a little bit. It was kind of like the occlusion that you would feel breathing air if you were breathing through a coffee stirrer, those little plastic coffee stirrers. So you could still get air back and forth easily, but it occluded it some. Well, my friend was elated. He was ecstatic. And he went to the FDA to get approval for this device. And he said, I want to tell you that this device or these two devices together are not equivalent to standard CPR. They're superior to standard CPR. One of the things I want you to understand when you look at FDA regulated trials is maybe you don't want to make such a big claim because the FDA said you don't have the data to claim that right now. An animal study is not enough, and they denied him. And so he had to actually go to Paris, France. And I went over there and watched them test this. Like I said, this was the first FDA trial I, ever went and observed, the FDA regulated trial. And in France, they showed that there was some superiority. And so he did eventually come back. It took years and years for him to get approval through the US FDA, though. And so anyway, I just throw this out there as a great story, but this is how cool things come about in medicine. So now we're going to pivot and we're going to talk about you and your family and your interest in research. And over Thanksgiving, you were sitting around the Thanksgiving table and you all were having chit chat and your sister was there and different family members and your sister, who's a paramedic, looked over at you and said, oh my gosh, the most terrible thing just happened. My partner actually in the back of the ambulance or the back of the box, as we call it, was recapping a needle, which I know is a no-no, and she accidentally stuck the needle in her finger. Oh my gosh, it was terrible with the patient's blood in there. And so she had to go and be tested. She had to take all these different prophylactic medications and everything. And it was terrible. And the medicine made her so sick. And the back of an ambulance is so bumpy. You go over all those potholes in the city, and it's just terrible. Well, your uncle, who happens to be, if I can get to the uncle, there we go, an orthopedic surgeon, said, oh, yeah, that is a huge problem in medicine. People get needle sticks not infrequently, and it is a big deal, and it's scary. And people, have to take these medicines and may still, get a disease from it, Hep C or something like that. And so he said, there was a TV show back in the 60s and maybe 70s, I can't remember. Anyway, it was called Star Trek. And I don't know if any of you all have ever watched Star Trek, but it was classic back then. And interestingly, a lot of the ideas that the producers and the writers put into Star Trek, they actually thought, oh my gosh, that's a very cool deal. And they actually became part of our day-to-day lives because, in part, they were first shown on Star Trek. Well, this is Dr. McCoy, the doctor for the USS Enterprise, which was the starship, and it was called a hypospray. And the hypospray was a device that would actually inject across the skin or transcutaneously these interchangeable drug ampules. Has anybody ever seen Star Trek? Give me a thumbs up. I want to, or a smiley face. It's very cool. Okay, I see at least one thumbs up. Oh, several. Cool. So you may remember the hypo spray. And so your uncle said if somebody could develop one of these, it would change medicine. Can you imagine if you could just change out these drug ampules and just boop, give it across the skin? It would be very cool. And he said not only would it change medicine, but it could be very financially lucrative. And many years ago, he said, the military actually worked on a device. The device that they worked on was for a jet hypodermic that gave across the skin inoculations for vaccines. They didn't have it for everything. And so basically, it just blew the vaccine into the arm. So what they would do is they would line them all up and just go boom, 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 and shoot them on the way down. And there was no needle to have to change, no, stick or anything like that. But which sounds like a great idea. However, I think you can see by this next picture that even though it was fast and safe, there was a little bit of a drawback to this thing, and the recruits did not like this. It hurt like the dickens. And so you thought, oh my gosh, Uncle, if we could just develop something like this, we might even win the Nobel Prize if you could do a whole bunch of different drugs and for a whole bunch of different indications. So you look up the different ways that the hypo spray was used in Star Trek with Dr. McCoy and the other subsequent doctors, and you kind of looked at the different devices that they had, and you saw how that he actually saved the captain with this hypo spray, and you were impressed. And so you decide that you're going to go to the lab and you're going to build a hypo spray device that you want to market. So, what is step one for your device? Step one for your device is to become an expert and become an expert in the way that different drug delivery systems and drugs can be used. So I'm going to show you all the different things that are possible, and you're going to understand why it's important to become an expert. So you can give meds sub-Q, which is subcutaneously, which is right below the skin, not in the muscle. You can give it SL. These are all medical terms that are used. Sublingual or under the tongue. You can give it IM or intramuscular. And that's probably what most of you all have gotten your vaccinations and shots. Most of them are IM, and they can be super painful. IV, through an IV, of course. PR, this is the one that nobody likes. This is per rectum or a suppository. That's another method. IO is something that you all may not be familiar with, but if we can't get IV access in a critical patient, then what we do is most frequently we take this thing that looks like a craftsman drill and we drill access into right below the kneecap in that knob under your kneecap, and we can give IV fluids and a bunch of different drugs, and it's only done in life-saving times, but it's another option that we have. Intranasal, like for fentanyl overdoses now, we're using Narcan intranasal. A lot of drugs have gone intranasal like that, but some of them, based on the drug properties, are not appropriate for intranasal. Transdermal, like patches for hormone replacement therapy and things like that, are also used. But this transdermal is very different than what is with the hypospray. And then the jet injection, like we saw with the military. So, why is it important to come up with a review article when you're doing this? It's because it provides background for things like grant funding or your IRB application and what you want to do when you pull together a review article that makes you the leading authority and perfect example in that lane within medicine is it explains the background and the need and the current state of the art. Where are we right now? So you would reference the jet injector and things, and if nothing's come out since then that would be the closest thing to what you're wanting to do right now. A review article or a deep dive in review of the literature also makes sure that you're on the right track and you're not working to duplicate prior failed experiments. And if you see prior failed experiments, there may be things that that you notice that you say, oh my gosh, if only they had looked at it in this way, I could reproduce this experiment in a preclinical way and I could make it better and potentially make it work. So it gives you that background so that you're not wasting your time and it sets you up for your grant funding. So you look at a number of different ways to potentially make a hypo spray. You choose one and you go to the lab and you start testing it. And here is your prototype of your star spray that you have. You give a nod to Star Trek and you call it star spray instead of hypo spray. So, in order to get an FDA IDE, after you've done preclinical research, after you've done your lab testing, you've looked at pharmacokinetics and pharmacodynamics and how this interacts with the actual skin that you're touching when you do this, you need to get an FDA IDE and you need some clinical input. You want to make your application very clinically relevant. And for the most part, particularly for an injection device or something like that, you do need an MD or a DO. And there are a lot of reasons for that, but you need somebody, you need to get that input. So you need to find a clinical partner. So how do you find a clinical partner? Well, you go and you present your bench translational research at a national clinical conference is one way to do that. There are a lot of ways to do that, but this is one way. And because you're excited about your results. And so 3 people come up and talk to you after your talk, and they're all enthusiastic. And I want you to think about which one of these 3 people you would choose in order to be your clinical partner. The first is an enthusiastic assistant professor at a big medical center that did a ton of bench research as an undergrad and as a med student, which is not uncommon in med school. They've never done a clinical trial yet. Okay, let's keep them in mind. The next person who comes up to you to talk is an associate professor who's conducted some original retrospective studies. So when I say retrospective studies, it may be a chart review. So they have access to the electronic medical record, and they may go back and say, out of all these people who had appendicitis, I separated out the people who got antibiotics versus the people who got surgery, and I looked at the difference in outcome. But I didn't randomize the people or do it prospectively. I only looked at it retrospectively. So he has not done prospective clinical research, but his chair feels like he needs a project in order to go from associate professor to full professor. So that's another person who talks to you. And then you have a very busy associate professor who has protected research time from the department and has an established research team and has training in clinical trials. Okay, let's see if you want to vote. Who votes for number 1, the enthusiastic assistant professor? Who votes for number 2, the associate professor who's conducted some research? And who votes for number 3, the very busy associate professor who has protected time and training? Well, my best guess would be to go with the very busy person. And the reason why is because the very busy person who has protected time, who already has a research team, and who has some training in clinical trials is probably going to be the most successful person at helping you formulate the things that you need to get done in the trial design and in the execution and things like that. So this is just provocative. I mean, this is not exactly how this is going to happen, but this is just provocative for you to think about what may be most helpful to you in your endeavors and in your work. You have to think of it when you're doing preclinical research or you're doing early clinical research already, and you want to step it up, or you want to get an IND, which is a FDA investigational new drug, or the IDE, which is the investigational device exemption. Um, Think of it like a marriage. Different people bring in different things, but both of you need to share the passion and the dream and make sure that you can set aside the adequate time and resources and have the expectation of ending up in the same place. I think you would need a person who also wanted to change current practice and who was excited about it and who was willing to get in and do the work. So, in order to get all of this launched, you need a research question. Well, here's how I like to think about research questions. My son and I were walking along at the ranch one day, and he picked up a rock a couple of 100 feet away from this tree, and he chucked that rock right into that hole that you can see, I think. I hope you can see on the left side of that tree. There's a big hole. And he looked at me and he said, "Mom," he said, "look how good I am. Look how strong my arm is. Look how accurate my aim is. I should be a pitcher for the Texas Rangers." And I said, "Now, son," I said, "I'm sorry, but I'm fully unimpressed because you could have hit that hole by chance alone. Now, if you had told me before you threw that rock that that was where you intended to put that rock, I would have been super impressed. I would have been so impressed." And the reason this is significant to research is because back in the day, people didn't pre-specify their endpoints. They had a good idea, they went and did their research, and then they dug through the data and found out what was interesting, and that's what they reported as their primary outcome. And so that got to be a real problem, kind of a data dredging thing to find the most uh, significant p value in everything that might have the most significance. And so what happened was they started clinicaltrials.gov. And clinicaltrials.gov said you have to pre-specify what you think your endpoint's going to be and whether you make that endpoint or not. It's also very, very important with the IRB because the IRB wants to know that you pre-specified this. So, what would your star spray research question be? Well, your thought is, does the star spray work? But that's really not specific enough because you don't really call out what work means. And so you've got to be more specific. So another rendition of this research question could be, is the star spray safe and feasible compared with standard drug injections? You're getting more in line with it because you're saying, is it safe and feasible? But it's still not really specific enough because there's no true scientific hypothesis to test. Okay, so we know the answer to this one. Is the star spray equivalent to or superior to current standard of care? We are not going to claim superiority at this point. We are going to say we hope it's equivalent to the current standard of care, even though we think it may be better. It's much easier to prove this equivalence than the superiority, and you don't want to have a problem with the FDA when you go to get approval from them to start your clinical trial. So a pilot research question or some rendition, a little more drilled down, might be, is the star spray safe and feasible in the clinical delivery of, and you can pick a drug, fentanyl, epinephrine for cardiac arrest and arrhythmias, nitroglycerin for heart failure, in an adult population in the pre-hospital setting. So you've said what you want to test. you haven't said the dose yet, but this is a pilot study, and that'll be within the protocol. You've said you've named some drugs. You can pick one of the drugs depending on the indication that you want. And you've said you're going to do this in the adult population in the pre-hospital setting. Now, for a pivotal study question, you need to drill down even a little more, maybe. Does 50 micrograms of fentanyl administered with the star spray, and I'm going to tell where I'm going to give it, in the right carotid triangle, which is close to a lot of big vessels right here, provides similar pain relief and ease of use. We've kind of looked at safety. You're always going to look at safety while you're testing something, but in the pre-hospital delivery, which means in the EMS system, of fentanyl. And then I've got my population here in healthy young adult patients with presumed isolated orthopedic injuries. So, like they're in a car wreck and they have a leg or an arm fracture, but they don't have a head injury where they can't talk to me, compared to an intravenous dose. So, this is the more detailed. You can see as our thought has moved along, we've gotten a little more specific and a little more detailed about this. And these aren't perfect examples, but it kind of gives you an idea of the thread that pulls through these in different phases and different thought processes. I'm going to shift a little bit to methods. And when I read a manuscript, one of the first things that I go to look at is the methods. And why are the methods so important? Let's say everybody on this call I sent out to make me a strawberry and a white cake. Well, here's a strawberry and white cake. Here's a strawberry and a white cake. And here's a rendition of a strawberry and white cake. They all followed my directions, but my directions weren't really specific. And one of the important things about research and when you report your methods is to make it a little bit like a recipe so that other people can follow it. It's very important that if people want to reproduce your research, that they can successfully reproduce it. And so it's very important for this replication, potential replication, which validates when all the data comes out of one lab on one new device or something, it it sometimes brings some criticism because there's a fear that it can't be reproduced in other labs, particularly in preclinical or in early pilot studies. It's important for clinicaltrials.gov because clinicaltrials.gov wants you to give some specifics. You don't have to give every specific in there, and I would caution you to try to give as little information to clinicaltrials.gov as possible because you have to end up reporting on all of this. But the Office of Research and Innovation has experts in this, and we are so happy that they're here because clinicaltrials.gov can be super challenging to deal with. It's also important for the study integrity to talk about your methods and for the FDA and for the IRB as well. So the IRB, a lot of people feel like the IRB is there to potentially prevent research from going on or to make it difficult, but that is not at all the case. The IRB is there to ensure a lot of different things, and they want to facilitate good, safe, wonderful research that is that represents you and your lab and the university well. And they want to make sure that the risks to participants are minimized, that the risks are reasonable in relation to the potential benefits, that the selection of people are equitable. So, for example, at UT Southwestern, where I still sit on the IRB, and at here as well, if there's a potential benefit to the research, then the consent forms need to be available in more languages than just English. Because if you don't have consent forms in other languages other than just English, then those other people may not be able to benefit from the potential medical intervention that you're studying. So, for example, at UT Southwestern, All the cancer studies have to be, the consent forms have to be fully translated into Spanish, and there need to even be short forms. I think UT Southwestern has about 40 different short forms in different languages that may be infrequently used, but can be used in addition to a translator with the English consent form. Uh, Informed consent is gotten appropriately and or appropriately waived for all the people, and it's well documented that the research protocol includes a plan for your data and safety monitoring, and that can be a data safety monitoring committee, a board, or if the risk is minimal, it can just be the PI and the study team. That the privacy and confidentiality of the people who participant is protected, and that if there are vulnerable participants like prisoners or people who are in other ways vulnerable, minors, things like that, potentially that they're well protected. So the IRB draws a lot of its standards from the Belmont Report, which looked at respects for persons, beneficence, and justice. But really, what the IRB is trying to do is protect the rights, safety, and welfare of the participants. So the IRB, when a study's submitted, they can approve the study flat out, which is pretty rare. I mean, almost very few get approved right off the bat. They can require some modification in order to get approval, and they'll tell you exactly what that is. And it can be a very minor modification, or it can be a major modification. They can actually flat out disapprove, which in all my years of serving on an IRB, I think I've seen one study disapproved. They can suspend a study if new information comes out, and that does happen sometimes. New information comes out and they need to change the consent form, and so the study may be suspended if there's new information about a drug or something like that. Or they can actually terminate the study, and that's also something that's super rare in the IRB world. So let's talk for a few minutes about the execution of clinical trials. I want to talk to you about some things that I have learned over the years in order to collaborate with people. And my undergraduate degree was in business. And so I like to think about running clinical trials in a business way in addition to a scientific way. And so you need to be a little bit of a salesman. You need to be a product expert, which a good salesman is. You need to be a cheerleader and get people excited about what you're doing, and which a good salesman is. You need to be a teacher or an instructor to be able to explain why what you're doing is important and how it can be accomplished. You need to be a great communicator so that people will enjoy listening to your presentations and get excited about it. And you need to be a motivator. You need to be a creative thinker and think outside the box. And it's important for all of us to be mentors, to mentor the next generation of researchers who are coming up. So just as someone hopefully has helped you or will continue to help you, you need to turn around and help others so that we can keep a awesome stream of research going. And it does take hard work. It takes a lot of hours. And so when you're working with a clinician, you need to keep your clinician motivated, in compliance, and definitely on track. So there will be manuals that get developed. There will be forms where information has to be taken down about participants and things. And a lot of times those forms that used to be on paper are now in REDCap, and we have incredible REDCap expertise within the Office of Research and Innovation. Educational material about what it is about your star spray. So it may be brochures that need to be approved by the IRB. It may be, there's just the sky's the limit with the things that you can do that they may want to put out in their clinics for participation in these studies. And the brochures can also be targeted actually to the providers. You need to be able to give training to these people, and you need to make frequent contact with your clinicians. that are the participant facing folks. And a lot of people think, oh my gosh, I launched my study. I put it out there. I gave them a brochure, but they don't touch base frequently. And so I think that that is one reason for slow enrollment, potentially. I think that working in the ER at Parkland Hospital, we had a lot of studies going on. And my fellow faculty would talk about a study in the faculty meeting, and they'd go, oh, yeah, we're trying to recruit this many people with heart failure for this drug or something like that. And I'd be sitting in the ER and I'd go, oh gosh, these three patients have heart failure. I wonder if any of them meet inclusion and exclusion criteria. And but I can't remember what it was, and I can't remember who was running that study. And oh well, I'm too busy. I can't figure out this right now because people are really sick and maybe dying. And so it's really important to keep frequent contact with clinicians that you're trying to get to help you recruit. Academic clinicians have a lot of pulls on their time. They have university committees if they're at a clinical site. Now, you can also use pay for play sites as well to do your research studies, but a lot of us have used the academic sites, and so that's kind of what I'm addressing here. They have constant training and recertification, teaching responsibilities, conference that they have to give and go to, their clinical duties, which can be a lot. They have journal club, and then they may even have their own research that they're working on. And so there are a lot of pulls. And so you just have to think about that in order to figure out when the best time to touch base with them is. Now, when I was starting my very first interventional clinical trial that was FDA regulated, for which I wrote an FDA IND and submitted it and got approval for, one of the sage, wise investigators there, because I was laying out my timeline. In 3 months, I'm going to do this. In 6 months, I'm going to do this. In 1 year, I'm going to have this finished, and this and this and this. He said, Dr. Wigginton, he said, I want to tell you something that was very hard learned by me, and that is when estimating how long it takes to get something done, double the number and increase the time unit by one. And that sounds kind of depressing and it sounds kind of tough. And it doesn't work if you have, a really long lead time. But his example was if you think something's going to take 3 days, it may take 6 weeks. If you think that something's going to take 1 week, it may take 2 months. And, so on and so on. So that was really important. And it really tempered my expectation with things. I think that another thing that's really important to do when you're launching a clinical trial is to identify your study stakeholders. So in this study of your star spray, you want to do this in a preclinical or a paramedic setting. And so you have to figure out what they think about doing this. Are they enthusiastic and excited about it? And if they're not, you need to get them that way and you need to get their input. For example, in the pre-hospital setting, these are like little mobile ICUs, these ambulances are, and they have a lot of equipment stored in a very tiny space. So you've got to make sure that what you package for them is as small as possible so that they can find a place to even put it on the ambulance. And that's just one of the things that you might not think about. People want EMS to test different things and they give them this big box or big suitcase and they're like, I can't test that. I can't even find a place to secure that within the ambulance. So think about the study stakeholders, the EMS people, the ER doctors who will be receiving these patients also, and pharmacy, things like that you'll need to work with. Now, this is not the best picture of this, but when I started my FDA study, I remembered that I couldn't remember anybody's clinical research and I couldn't remember who to call. So I took, if you can look at my little picture, a luggage tag that was heat sensitive, like you make these, like you put the piece of paper in with your name and everything, and then you run it through that heat element and it seals it. And then you take a punch, if it's not already punched, and you put a punch up here, and people can wear it on their name tags. Now, anybody in clinical medicine doesn't go anywhere without a name tag because you can't get in and out the doors. People have to know who you are. And so what I did was I put the basic inclusion and exclusion criteria, inclusion criteria on one side, exclusion criteria on the other side. I put my name, my phone number, and the name of the study. And I said, call anytime you have a question or you identify somebody. Oh my gosh, I had never seen anybody do that before. And it made a huge difference because then people could wear it on their badge. I made it myself. It cost nothing almost. And they could wear it behind their badge and they had it with them all the time. And so when they came to a person who they thought, gosh, this might be somebody for somebody's study, they said, oh, yeah, this person meets those criteria. It was Dr. Wigginton. I'm going to call her. And so it's just cheap and it's easy and it's effective. So don't forget about that. So I want you to approach a clinical trial with as good a money bank as you can put together and don't cut corners with your costs, but make sure you're fiscally responsible because money's always hard to get. I want to say that I'm excited to have talked to you all today because sometimes people with incredible ideas are sitting around thinking about them, but they don't realize that they're the missing puzzle piece to solving the puzzle. And I want to challenge you all to think about this different facets in the area that you work and how you can be the person to solve that puzzle. Another thing that was told to me, and the very first research grant that I got was from one of our Nobel laureates at UT Southwestern, and he is not living anymore. But he basically said, Jane, he said, I want you to understand that historically, young, unfettered minds make the most profound contributions. He said, you look at Nobel laureates, and these Nobel laureates didn't come up with these ideas when they were old. They got the prize when they were old, but it was based on the work that they did when they were younger and they thought outside the box and they were excited and enthusiastic. And even though I'm not young anymore, I try to be way unfettered and I try to be enthusiastic and excited and I try to always think outside the box in order to put forth the most exciting, the most impactful research that I can pull together. And so I would challenge all of you all to have unfettered minds and make these incredible, incredible contributions. So I want to let you know that I want you to look at the new Clinical and Translational Research Center. I would refer you to that because we want to help you in your journey. The Office of Research and Innovation also wants to help you. The IRB wants to help you. But this is something that you can come and have consultations or ask questions of. And we're working to bring the pieces together. You can see in this picture that was beautifully done by the Office of Research and Innovation, that it shows the pieces being put into place in this beautiful mosaic. And that's what we really hope to do. We hope to be a catalyst. And so just come at us with questions. We're here. You can look it up, CTRC at UT Dallas. And I'd like to see if there are any questions right now.
We do have one question in the chat, Dr. Wigginton. It says, can you please tell us about a clinical trial or collaboration in your experience that stood out to you the most and what was so interesting about it?
Oh my goodness. Yeah, I think that, well, I have two. And I'm going to tell you about them real quickly. One is working through the Resuscitation Outcomes Consortium, which was a between 10 and 12 site, depending on the year, look at doing resuscitation research in cardiac arrest and in trauma patients. And the survival rate, these were really severe cases, and the survival rate was so low that we had to enroll at all sites up to 100,000 people in order to answer the question. And so it was very interesting to see how these multi-site groups who were so passionate about this. I mean, these were the leaders in the field, and the NIH funded it, and it was just incredible. And I learned so very much about that. So many questions were answered, so much data was collected. I mean, we learned that even before we could do cardiac arrest research, that we had to go out and teach people how to do optimal CPR in order to make sure that the interventions were tested accurately. So that was very cool. And then one of the other clinical trials that really, really speaks to me is the Texas Biomedical Device Center. We are currently testing a very small, about the size of your little fingernail, vagus nerve stimulation device that's implanted for spinal cord injury and for stroke participants and for people with PTSD. And we've finished the pilot trials on all of these, and the results have been amazing. But it's the first time I worked on an investigational new device, and it has just been very cool to see an academic center pull together a implantable clinical trial without using somebody else. I mean, we used surgeons at Baylor. I won't say without using somebody else, but it's not like we got the idea and we gave it to a device manufacturer and they just took it and ran with it. I mean, this is something that we have shepherded, and it's been an incredible experience. And I hope that that experience can translate into help for a lot of other people here at the university.
Awesome. Thank you for that response, Dr. Wigginton. And another question says, excuse me, can you tell us a little more about navigating interactions with FDA during study planning?
Yes. So I think that if you want to navigate questions with the FDA, you just need to pick up the phone and call them. When I was doing my first FDA IDE, I didn't have any experience and I didn't really have a mentor. And I picked up the phone and called them, and it feels a lot like that they really do want to help. And uh, particularly if it's an investigator initiated, because they know that, if they're dealing with big pharma or a big company, they know that they have all kinds of people to do all kinds of things, and they have a lot of pathways already set. But an investigator initiated IDE is something that is best talked to with them. Now, I still have all my IDEs, and we have INDs through the Texas Biomedical Device Center. And if anyone is interested in looking at what one looked like when I submitted mine, people think that these INDs or IDEs are this thick. I don't know if you can see my fingers, but mine are only about this thick. And it was the repurposing of a drug for a new use that it had never been used for. But it kind of gives you an idea of what they're looking for. And I think that we can be a resource at the CTRC also to help. But I would absolutely pick up the phone and call and talk to them. I don't know the order because I don't know exactly what it is you're wanting to study. You could always come to us first, and then we could talk about you chatting with the FDA. But I have found them to be incredibly helpful, and even I'm not going to say a champion of research necessarily, because they have to hold equipoise about these things. But I do think that you can sense their enthusiasm for practice changing innovation. I mean, they have their rules, and they are very, very dedicated to sticking by their rules. But I think that they can be super helpful.
Excellent. Thank you. Are there any other questions from the audience? Please feel free to turn on your microphone and ask your question. Are there any other questions? All right. If there are no more questions, excuse me, my voice keeps going out on me. If there are no more questions, we're going to go ahead and close out with, first of all, by giving a huge thank you to Dr. Jane Wigginton for not just sharing your knowledge, but for inspiring us all with your passion and expertise. Thank you, Dr. Wigginton.
Oh, thank you so
We
much.
appreciate the hard work that goes into gathering this valuable information and sharing it with us. So let's all send some virtual applause and appreciation to Dr. Wigginton. Audience, you guys know I'm going to thank you as well. We always appreciate you all being here with us. So I'm going to close out by saying we have another session coming up on Wednesday, March 26th, the second in this session, the clinical trial series, where Dr. Seth Hayes will present. We hope you all will join us for that. Keep up with what's happening in the Office of Research and Innovation. And Dr. Wigginton, I'm going to grab the screen share here because you guys know I'm always going to encourage you to follow us on our social media. Please do that to keep up with all the great things that are happening in the Office of Research and Innovation. Now, are you interested in a broader impacts toolkit consultation? During the one-on-one consultation, a broader impacts professional will walk you through a free toolkit that you can use to develop a competitive broader impacts plan for your NSF career proposal. The broader impacts professional can also assist you with developing a long-term relationship with community partners to satisfy your proposed activities. So I dropped a link in the chat. You guys can use that link to sign up with Ms. Tiffany Willoughby by emailing her at tiffany.willoughby@utdallas.edu. And last but certainly not least, we love to hear from you guys. We love your feedback and we appreciate and value it. So please take a moment to scan the QR code and share your thoughts with us. Your input goes a long way. We use that to determine what kind of information we can bring to you guys, what's valuable for you. So please take a minute to scan that QR code and share that information with us. We will stick around for a few minutes. If you guys have any other questions, feel free to stick around and we can answer any questions you have. If not, feel free to log off. You guys know I don't like leaving people behind, so feel free to log off if you don't have any questions. Thank you all for being here today.